Hi, everyone. It's a real pleasure to be with you today, virtually, unfortunately, to tell you a bit about what we've been working on this past year with respect to our slice cultures and strain-specific pathology. So first, I'd like to thank the sponsors and all of you really for the gift you give to keep this research going. So I want to recognize Katie Pohl Dopuric Memorial Research Grant, the Jeffrey A. Smith Memorial Research Grant, the Robert J. Esposito Memorial Research Grant, Kendall Palmer Memorial Grant, and of course the Strides for CJD grant. Now, today what I'd like to talk a bit about is variation, and that specifically refers to how patients present with CJD. You may be aware that patients present with a variety of symptoms. Sometimes it's prominent visual symptoms. Sometimes it's just incoordination. Sometimes people have dementia and cognitive changes early in disease, sometimes late. And this can complicate the diagnosis, even delay the diagnosis at times. But my question today is, why is there variation? In neurology, we say it's location. The brain region that is affected dictates the symptoms that appear. So if you have a stroke in your occipital lobe, you're going to have visual loss in the opposite side. And we do know that prion diseases target different brain regions. But again, why would the prion target a different brain region? We know that prions come in different flavors, different strains, sizes, shape. You know that there's the normal form of the prion protein shown here, and when it misfolds, it takes on a different shape. There's still a lot of debate in the field about what that shape is. There's some recent models that have been proposed to try to describe that shape. And even here, we know that it can't just be one shape, but there are a variety of shapes, each associated with a different strain. More than shape, it's also size that differs. For example, shown here is a recent work published by my group showing size distributions of these clumps of prions. In the blue, you can see that this particular strain has more smaller aggregates than some of the other strains. And we think this too can correlate a bit with phenotype. That is how patients present. What is the clinical correlate? So how might this work? So here's my poor man's drawing of a brain. And let's think about how prions might target different parts of the brain. So let's make an assumption that prions could potentially target any part of the brain. Why would you have disease or phenotype coming from one area more than another? Well, it's possible that certain areas of the brain just aren't as efficient at replicating, so they don't build up as much prion, therefore there's not as much damage and you don't have symptoms from that area of the brain. Another possibility is that certain brain areas might still accumulate the prions, but somehow they're less prone to damage from that particular type of prion. So we wanted to think about how we could test this question, and we use something called the Prion Organotypic slice culture assay for this purpose. You can think of it like prion disease in a dish. Basically, we take the mouse brain, we slice it up, and we put it in a dish. And then we can sprinkle prions on top of it of any strain we want and watch how the disease develops in the dish itself. And this is just thought you might be interested. This is actually what it looks like by naked eye when we culture it. And at the bottom, you can see cerebellar slice culture. That's the back part of the brain. And that's how this assay was initially developed by Adriano Aguzzi's group. And this is on the bottom, you see what cerebellum looks like. On the top is what we've been doing now is slicing the front part of the brain, basically the rest of the brain, in coronal slices. So slicing it as if you're slicing your face. And we wanted to do this because we're very interested in the different brain regions and how they are targeted with prions. So this is what they look like just in the dish. This is what they look like when you label them, the cells, with different colors under a confocal microscope. So here's an example of a zoom in on a slice. The red is the neurons, blue are microglia, the cyan are astrocytes, and the green is actually the prion protein. And if we focus more just on the prion protein, in this slide in green, you can see the clumps, I think, of prion protein that are building up in the disease. And interestingly, in blue in this image are the microglia. And at the very end of the talk, I'm going to mention microglia a bit more. So in these slice cultures, where are the prions accumulating? And do we see differences with different strains? If you look closely, here's a sort of a screenshot of a number of strains that have been looked at. At the top is our control, where you don't see any signal. ME7 is a particular strain where, again, it's very weak. We don't think we get a lot of infection from this particular strain. But as you go down in RML, 22L, and 87V, I hope you can appreciate that the pattern we're seeing is very different and seems to relate to which strain is involved. This had previously been looked at in cerebellum, but this is the first time we've documented this difference in these coronal slices. The other question is, where is cell death occurring in these slices? Is that also different depending on strain? Well, here's a few examples of what the pattern of neurons looks like. So in the top are some control uninfected slices, and bottom is an example of RML. And you can see that here too, it's biology, we have some variation, but in general, we see less neurons in the infected slices, as you might expect. But what we found particularly interesting was the region that is lost in most of the slices. So if you look at the top, you can see right here, hopefully, this is actually the hippocampus. And in our infected cultures, if you look in the same area, we don't see those hippocampal neurons. This dentate gyrus seems to be gone. And in fact, if we quantify it, we can see that while maybe 10% of our control slices don't have any hippocampus left, the infected samples have a great deal of dentate gyrus loss. Now, that suggests that prion infection, actually regardless of strain, seems to target the hippocampal neurons in culture. But that also tells us it's not necessarily specific. It's not telling us why one strain would cause a particular phenotype. So then we thought, could it be that it's about the spread within the slice itself? So instead of assuming that the entire brain is coated all at once with prions, as we would normally do in our slice culture, we immerse the entire brain in prions. Perhaps there are certain areas of the brain where it spreads more efficiently than other areas of the brain. Is there a way we could test that in our dish? This is where we thought about doing wire experiments. Essentially, we cultured the slice as usual, but then we took wires and incubated them in the prions, dried them, and then put them on top of the culture to see if that could target specific brain regions to start the infectious process. So here's an example of what it actually looks like. If you put the wires on top of the slices, we had visions originally of stabbing the slices directly, but this is a much magnified image and it's very challenging to manipulate these particular wires. So we did this to start. In the middle panel is simply a picture of what the slices look like after we transfer them to a membrane without any labeling or staining. Then we digest away all the protein except for the prions, and that's what's in the right panel. So you can see sort of those dark splotches. Those are the prions after 30 days of infection. And I hope you can appreciate that they sort of line up along where the wires are. I'll show you another example here, which might be a bit clearer. Again, on the right are the dishes with the wires placed, and immediately beside it to the left, you can see these streaks of prions along where the wires were. And I should say these wires were removed 7 days before the actual transfer to membrane was occurred. We also did some controls, of course, some uninfected cultures, and even some slices exposed to an infected wire, but slices that themselves don't express prion protein, so can't propagate any of the infectivity. And there we didn't see any signal. So this was at day 30. We thought, well, I wonder if this is the start of infection in these specific regions, but will it then progress fully to the entire slice or not? So we've just recently done some day 60 examples, again, exposed to wire, and we have found that in this case, it does start to resemble the pattern where we infected it just by immersing the whole slice. So you may recall this is an RML where we infected without the wire, and the histoblot beside it shows infection with the wire, but given time to spread through the whole brain. So we are still doing these studies. We're starting to do other strains now. And it does appear that at the moment, focal infection at given sites still spreads to the same strain distribution. But of course, what about human strains? So we've done some of these experiments with human strains as well, using a mouse that expresses human PRP as slice culture. So our early experiments were very positive because we were seeing this detection of abnormal prion protein in a number of different strains of human prions in culture after 43 days. So over the past months, well, suffice to say, COVID has put a little bit of a limitation on what we've been able to do, but we got in some really important controls. So we took mice that don't express prion protein, and we did the same control experiments and added CJD brain and looked for any signal. Of course, these slices should not propagate because they don't contain PRP. But we found trace amounts, shown here at day 14, day 28, day 35, trace amounts of prions left in those cultures, telling us that we haven't fully gotten rid of the initial inoculum. Now, if we compare it to the infection that we see in the actual TG human mice, it's not as strong. So we do think we're propagating something, but it's challenging because now we think there's a bit of residual in addition. And we checked the other strains as well, and essentially found very similar findings. So what do we do about this? Well, this is where we turn to the wires, because here we can just put a very specific targeted inoculum through a wire without spreading it throughout the entire dish. We actually tested a whole bunch of different wires, stainless steel, nickel, copper, etc., and really found that steel and actually acupuncture pins were the only ones that wouldn't oxidize. And we're hoping that acupuncture pins could actually be more precise. Instead of laying them across the slice, we could actually stab the slice. So here I'm showing you that we can bind the prions to the wires, and then we put them into culture. Unfortunately, after 58 days, this blank lane here, this says we did not detect infection after skewering our slices. And when we went back and looked at the pins that we put into those slices, we couldn't find the prions on the pins anymore. In fact, we looked at a higher contrast, and really, if there's anything, it's really, really low level. So where are we now? It looks like CJD prions may be propagating in culture, but we've got that residual leftover from the initial infection, making it hard to distinguish new infection from old. So we want a more precise way to infect them, and hopefully a more targeted way. So when we move to wires, it makes sense because we can target a particular area, there won't be residual anywhere, and they're working quite well for our rodent-adapted scrapie. But so far, it seems like the pins themselves lose the CJD prions. So as often happens in science, it's not so much that you make lemonade out of lemons, but the question coming from an experiment sometimes leads to new questions. And our question now is, where did they go? Where did the prions go? So let's go back and think about this. I was suggesting that perhaps regions for specificity involved easier propagation or spread, but maybe some brain areas are better able to clear prions. So that is now leading us into the microglia. These are these resident macrophages. They're a sort of inflammatory cell, and there's a lot of literature growing on that they can be either good or bad in the context of disease. We know if you suppress them in culture, you can increase infection rates. So perhaps this is our next step in human cultures. And we want to ask, do microglia have a strain preference? Could they be the reason that certain strains preferentially target certain areas of the brain because they can be taken up by macrophages in those areas of the brain? So that is the story to be continued. And I want to, in the last few seconds, thank, of course, the people in my lab who've done the work. Here's a picture of us all pre-COVID, when I still had short hair and we could actually be together. Satish has done a lot of the work on the CJD cultures. Haley is another culture guru, as is Grant. And Leo has done a lot of work on analyzing the aggregate sizes. So with that, I will wrap up.